Okay, next up on our rough cut series, we're going to talk about Vdj recombination. That's V, and then parentheses D, J recombination. And so what Vdj recombination is, this is a way that that B cells generate new antibodies, um, or generate antibodies at all. So now, this is not directly involved in evolution because B cells are not in the germline, and so the things that they do are not directly applicable to evolution. That is, the mutations and the recombination that occurs here doesn't get passed on to progeny. But nonetheless, I think it's very good because it demonstrates the competency of cells in managing their own DNA and managing their own mutations and recombinations, and that there's structure and function here that's it's not just haphazard. It shows that there really is a very strong competency of the cell within managing its own DNA. So, just kind of a super quick, intuitive look at the immune system. So when you have an antibody, we usually draw antibodies kind of like this. Um, and so in here, so this is what an antibody looks like. And so you have there's kind of two main parts of it. So this part attaches to the antigen. It's kind of a grabby part. So this is the grabber. Okay. So this part signals the immune system. And this, in general, is known as the variable region, although there's also a more specific part that's variable. And then this part is known as the constant region. Okay, so the constant region. So if you think about this, the reason why the top part is variable and the bottom part is constant should be pretty obvious. The top part has to connect to all sorts of different organisms and antigens, and the bottom part has to signal. And so the signaling is fairly uniform in the immune system. Okay, so now your body has millions of different antibodies, but it only has a couple hundred genes for them. So now, so how does it produce so many antibodies from just a couple hundred genes? Well, the answer is that it basically does. If you remember, kind of the story of interchangeable parts from high school history class, where I think it was Eli Whitney. He was able to put together rifles that he could just pick up um, a trigger from one bin and a barrel from another, and I don't know all the parts of a rifle, but anyway, he picks out each part from a bin, puts it together, and has a functioning rifle. Okay, and this is in contrast to beforehand. All the rifles had to be handmade, and the parts didn't interlock well. Well, imagine that, but instead of having barrels of parts where they're all identical, imagine that they all still interlocked, but they were all different. So there's a box of different triggers, a box of different barrels, a box of different I don't know, different shoulder mounts. So all sorts of they all interlocked together, but they were all different. So you could actually get millions of guns from a couple hundred different parts by picking one part out of each barrel. And that's what happens with Vdj recombination, is that you basically have in your DNA, you've got a bunch of different genes called V genes, and you have a bunch of different genes called D genes, and you have a bunch of different genes called J genes. And when a B cell is maturing, what it does is it basically picks one of these from each bucket and combines it together and to have a Vdj. And so the technical names, so this is a variable, this is diversity, and this is joining. Okay, and this whole thing together can be called variety of different things depending on who you read. This is the variable region, or the CDR, which is the complementary determining region. So it determines how it complements to the antigen. So basically, when a B cell is maturing, it picks one of these from each bucket and pulls it together to make a single variable region. Then there's a constant region, which is the region that that tells it how to signal the rest of the immune system. And there's more than one constant region, but each constant, there's only a couple of them, and each one kind of has a specific type of reaction that it triggers, like is it an allergic reaction, that sort of thing. And so the VDJ gets joined with a constant region. And so that generates a gene that's now the B cell's immunoglobulin gene that it uses for that particular cell. And there's millions of cells, and so each cell is going to basically choose one of these at random, one of these at random, and one of these at random. And by doing that, it gets a huge diversity of immunoglobulin genes from just a handful of individual genes. And the mechanism for this is pretty fascinating. There's two main genes, or two main proteins that do this: RAG1 and RAG2. Don't know what I did there. Undo. RAG1 and RAG2 that cause this recombination to occur. And so basically there are signal sequences that help these guys do their business. So these are called recombination signal sequences. And basically, so this has a 7 base pair sequence, then a spacer, then a 9 base pair sequence. Now the spacer is either 12 base pairs or 23 base pairs, and the reason why it has this difference is that that way it knows that when it's joining two segments, that it should join a segment that has a 12 base pair spacer to a segment that has a 23 base pair segment, and so that way that it knows that it's joining the right thing to the right thing, because you don't want to join a V region to a V region, or a V region to a J region. So by having this, some of them are spaced by 12 base pairs, some of them are spaced by 23 base pairs, and that difference allows it to know that it's joining to the correct thing. And so the 7 base pairs and the 9 base pairs are it's a highly conserved sequence, which means that it's basically identical. So that's the 7 base pair sequence, and that's the 9 base pair sequence. And while there's some slight variation, they're pretty typical. The spacer region, for the most part, it looks like it is just structural, and that there's not a lot that the spacer actually does, although there's some evidence that the spacer region may have some effect. So it's unclear whether the specific sequence matters or not. But the spacing matters a whole lot. So anyway, so by having these specific markers, it shows the RAG1 and RAG2 genes where to cleave to and to join together. Basically, it basically pulls together the DNA and cuts out the intervening DNA and joins it together into a new gene. In fact, additionally, there's also some evidence that there's additional modifications that can be made as well. But on the whole, it works by basically pulling two pieces of DNA together and then lopping off the intervening DNA. So the Wikipedia article on this is pretty good. So here you can see the DNA being pulled in. The article that I've always gone to, it's kind of dated now. Although my knowledge of this whole area is kind of dated. I haven't read strongly in it for a number of years. Is this paper on PLOS biology, Vdj recombination, and the evolution of the adaptive immune system? They talk about a lot of interesting features of it. And one other thing to note is that as the body changes its response to different antigens, that constant region can be switched in what's known as class switch recombination. So the body, it knows, it's got information on how to join these different pieces together into a new gene. It has information on which parts are the constant region that needs to be switched out as the depending on what the needs of the body are. So you have all these mutations that are occurring that are very, very highly regulated. And it's a fascinating system. But that's not the only thing. Additionally, if you have an antigen that you don't have an antibody for, your body undergoes what's called somatic hypermutation. And that's where it basically mutates the immunoglobulin gene to find alternatives. So let's go back to our little drawing board here. So basically what happens is, so imagine your genome, you've got about 3 billion base pairs. So 3 billion base pairs. Okay. Well, the antibody, I think it's about 1,200 base pairs. And that's not even to scale. So 1,200 base pairs. Okay. Now the interesting thing is that when your body needs a new antibody, it performs mutations on an existing antibody. So it skips over all of the other 3 billion base pairs. It skips all these and does not mutate them. All the mutations happen in this little region that is the immunoglobulin gene. So all the base pairs are focused on the correct gene. Not only that, again, we've got, remember, there's the constant region that signals, and then there's the variable region that grabs. Okay. So now if you think about it, if you've got a new antigen, you don't need to change the signaling, because if you modify the signaling, then the rest of the cellular machinery wouldn't know how to react. So modifying the machinery would be bad. The signaling machinery would be bad. What you want to modify is the grabbing machinery. And that's actually what happens, is that within the gene, all of the mutations occur in the variable region. So there's actually a non-coding DNA sequence that kind of targets where these mutations occur. And there's a chemical called AID, which is used to trigger the mutations. but the mutations happen almost exclusively in this region. And so out of 1,200 base pairs, I think it's around 600 to 1,000 that actually get mutated, and the constant region is basically left alone. So anyway, you have this really fascinating system where, again, the cell knows how to manipulate itself, and it's very competent in doing so. It has some great machinery in doing so. Another thing to note, which I think I've noted in one of the previous videos, is that sometimes one of the things that causes cancers is when a base pair is mutated so that it looks like a target. And so there's lots of cancers that are caused by a single base pair mutation that makes something look like a cleavage site for Vdj recombination. Like it makes it look like one of these recombination signal sequences. And so anyway, that's the so these individual mutations that are problematic have cascading effects. And so anyway, so on the whole, there's information that's prior established in the genome that helps it know where to mutate, where to cut, where to glue. And if that gets messed up, then it no longer knows where to cut, where to glue, and how to do these manipulations. So anyway, so that's Vdj recombination. Just a super quick overview, but hopefully is helpful. Again, I'd go back and look at these recombination signal sequences, just because I think taking a look at, I think it'd be interesting to take a catalog of all the different recombination type sequences that people had and how they functioned. So anyway, hope that was helpful.